Welcome back to Well, That's Interesting. It's been too long, so great to see them back in the news edition. Today's episode 286, rare confirmed sighting of jellyfish hunting, and finally, a study about salmon on cocaine. My friends, the last time we spoke about jellyfish was way back in October of 2025 during episode 254. It was at that time we grasped our baguettes and traveled to northern France, heading for a beige behemoth called the Gravelines nuclear power plant, who was recently in the news. Thanks to a spectacular swarm of jellyfish, 4 of the 6 reactors belonging to Western Europe's most powerful nuclear power plant went into automatic shutdown after all those gelatinous bodies clogged filter drums designed to remove debris as they take in cooling water from nearby seas. The reactors were simply overwhelmed by their numbers, and we were overwhelmed as we learned this wasn't the first time such a thing has happened. Oh, no. We learned of 7. Yeah, I'll say that again. 7 separate instances of jellyfish passively meandering into nuclear power plants worldwide and shutting them down. As to how this can keep happening, have a listen if you haven't. Today, in the first half of the show, jellyfish are back in the news, and this time, their destruction has nothing to do with chance or from going with the flow. For the first time, University of Southern Denmark biologists have shown that certain jellyfish capture and eat swarming polychaetes at certain times of the day at a certain season. We will meet the team that dedicated a year of research proving that not 1, but 2 species of jellies exhibit hunting behavior. We will chomp into their study and the number of jellyfish dissected. We will look upon what was found inside and run some tests to prove that these species are taking advantage of polychaete reproduction. We will learn what the fuck a polychaete is, and we will learn more about their sexy times than necessary. So, what does it mean that a brainless blob can hunt? We will answer that too. Then after the break, the last time we spoke about non-human creatures on cocaine was way back in August of 2025 during episode 249, when researchers gave drugs to spiders just to see what would happen. That was the title of the show, and it followed episode 246 when we learned about a study with harbor seals playing video games, which was adorable, and another study with fruit flies on cocaine, which wasn't. And we learned why all of this matters. Indeed, we have covered animals on controlled benders at least 5 times, and each of those studies were performed to help figure out how illicit drugs ending up in our waterways affect the health and behavior of innocent aquatic bystanders. Not only did this research have drugs in common, but all of these experiments were performed in lab settings, which do not necessarily reflect the complexity of natural environments. What if, perchance, we took some young, impressionable salmon and gave them, say, a slow-release chemical implant filled with cocaine, or its metabolite, benzoylecgonine, and released them into a large Swedish lake and tracked their behavior over the next several months? Well, I have great news. Marcus Michelangeli of Griffith University and his colleagues went ahead and did such a thing. Oh yes, we will crack open their experiment and rub the results on our teeth. What I mean to say is that we will meet the fish, the lake, and the metabolite, and we will see what happens when all of these 3 get together. By the way, young salmon are called fries, and these fries are loaded. That was a terrible pun, and hi, I'm Jill Chacha. And if this is your first time listening, welcome to the flock, my straight-laced business goose. To begin, I'm going to ask you to close your eyes and to reach back, way back into the marshy bog of memories, and to pluck from it that one summer night where within the waters of a fjord off the coast of Denmark, your body exploded like a piñata, releasing eggs, sperm, or even both into the surrounding area, all in hopes that they would smush together with others of the same species. Now, at this point, I'm sure you opened your eyes and said, Jill, what the fuck? Never has such a thing happened to me. Well, I know, and that's because you are human. I was actually addressing all the polychaetes in the audience, who, if they had them, would nod their heads in blissful recollection. Let's meet polychaetes, shall we? Because I know you're dying to know what exactly is going on here. Luckily for us, we don't have to travel very far to find one of the 10,000, yes, 10,000 species known to science. All you have to do is head on over to any of the world's oceans, stick your hand in, and you may just pet a polychaete, or a marine worm, or even better, thanks to the added detail, a bristle worm. Now, I said that there were about 10,000 known to us, and I have to say there's an equal number of varying characteristics depending on the species you pet. For example, some are less than 4 inches in length, although others range from 4/10 of an inch to 10 feet long. Their heads may include 2 to 4 pairs of eyes, although some species are completely eyeless. Some are brightly colored, and may be iridescent or even luminescent. Some prefer to crawl along the bottom of the sea floor, while others burrow and stay put. Or some like to swim to various locations. Perhaps the most famous marine worm of all is the tube worm, which you've definitely seen if you've ever watched a nature program about our oceans. These build their own apartments by anchoring their tail to an underwater surface. Here, around their bodies, they secrete a mineral that hardens into a tube, into which it can withdraw its entire body. This is extremely helpful, especially with today's cost of living. Despite this chaotic mixture of features, they do have some things in common. Their bodies are segmented, and along each segment are fleshy protrusions called parapodia, or bristles. Adults of 2 species, Alida succinea and Platanires dumerili, nailed it, they use bristles to leave their burrows, swimming upwards to spawn during warm summer nights around the full moon. During the last moments of their lifespan, they take this journey into open water, hoping against hope that they will find love. Now, this may sound extremely relatable until you get to the next part. Once thousands have gathered, every one of them will explode. Yeah, nature's plan here contains highly specific elements like summer, nighttime, the full moon, until you get to the end where they just explode and hope for the best. Anyway, that's how it goes. Millions of sex cells will combine to form future marine worms, but the remaining candy, if you will, becomes the staple diet of carnivores who show up to the party right on time, and animals you'd expect, like trout and other predatory fish. But what if I told you there are 2 other creatures who have this written down in their calendars, and they have no brain, heart, or eyes at all? You might just be saying to yourself, Jill, that sounds crazy, but honestly, look at what we're talking about already, so I guess that makes sense. But please, prove it to me. Okay, my delicious business goose, I will. Join me, will you? Off the shores of the inner Danish estuarine complex of Kittermind Fjord and Kirting Nor, between the months of August 2022 and July of 2023. And for my fellow geographically challenged Americans who are like, what the fuck is a fjord? And did you say ketamine? No, I said Kirtemund. And if you imagine Germany wearing a tiny triangular hat, that hat is Denmark. To the southeast is a small protruding bulbous piece of land whose upper right corner has been carved out by 2 connecting bodies of water, the Kirtemund Fjord and Kirting Nor, which get their supply of water from a narrow channel connected to the open sea. And all of this connection means there are fun, important facts that I need to drop on you. According to the study we're consuming right the fuck now, revealingly titled Polychaete Capture by Native Jellyfish and Invasive Ctenophore reveals a novel benthic pelagic trophic link. 1. This system exhibits a wild fluctuation in seasonal temperature. The water temperature can range from 0 to 25 degrees Celsius, or from 32 to 77 degrees Fahrenheit. 2. The salinity of the water does just the same, jumping from 10 grams of salt per 1,000 grams of water to 25. 3. Having a main line to the open ocean while being surrounded by rolling mountains and agriculture means that our fjord and Nor here are eotropic. which is a fancy way of saying tasty good shit rolls right into it. And I mean tasty good shit literally. Runoff containing fertilizers, sewage, or animal waste introduces excess nitrogen and phosphorus into the water. These nutrients feed aquatic plants and algae. And you know who loves to eat microbes, algae, and seaweed? Elida succinia and Platyneres dumurali, the two marine worms we just spoke about. The ones who burst at the seams on warm summer nights under a full moon. They call these waters home all year round, and it's their scheduled reproductive extravaganza that brings all the omnivores to the yard. And as such, all the commotion also happens to gain the attention of curious biologists, including biologist and postdoc Hannah Yo, biologists Laura Ferreira, Eric Christensen, Jamila Javidpour, all from the University of Southern Denmark, and Anders Garm from UCPH. This A-team of busybodies couldn't help but notice, among all the creatures who arrived, two stood out like sore thumbs. The common moon jelly, called Aurelia aurita, and the invasive comb jelly Nimiopsis ledei. Nailed it. And if you had listened to episode 254, the one about the jellyfish overpowering a French nuclear power plant, the moon jelly was a suspect, but not the culprit. And it was high on the list because they are just so goddamn common. They're found throughout the world's oceans in the northern hemisphere. But don't worry about your limbs, my northern business geese, because instead of long, tailing stingers, they have a fringe of about 1,000 to 1,200 very short, delicate tentacles. Their bell, or cap, is about the size of a dinner plate, and unfortunately, they're most recognizable by one very distinct feature that's very much in your face. Or rather, 4 of them. As described by wildlifetrust.org, quote, 4 circles are visible through their translucent white bell. And these 4 circles are gonads, the reproductive organs located at the bottom of the stomach, and they are normally purple in color, end quote. Yeah. These animals are 95% water, and you can see right through them, except for their bright purple gonads. What can I say? Moving on. The comb jelly, Nimiopsis ledei. It's native to the east coast of North and South America, and it also goes by the nickname the sea walnut. I know, but don't let that cuteness fool you. And don't let the word jelly fool you either. They're not actually jellyfish, but actinophore or comb jelly. And unlike cnidarian jellyfish, they lack stinging cells entirely. And that's the least wild thing about them. As reported by the Marine Biological Laboratory, the Virginia Institute of Marine Sciences, and the Smithsonian, they capture tiny zooplankton, fish eggs, and larvae using sticky, mucus-covered cells called colloblasts, rather than stingers. They have 8 rows of vibrating hair-like cilia, or combs, that they use for swimming. And when lights hit these rows, they flash pulsing rainbow-like colors. They are simultaneous hermaphrodites that can self-fertilize. A single adult can release thousands of eggs daily, and they reach maturity in as little as 2 weeks. They also possess a transient anus that only appears during defecation. Yeah, they can spontaneously form a butthole anywhere on their body, and I'm sure that fact distracted you from the one right before it, that they reproduce at insanely high rates and mature in just 14 days. And that may have also distracted you from the fact that they are native to the east coast of North and South America. So one must ask, how did they get to a fjord off southern Denmark? Well, they hitched a ride inside ballast water. Now what the fuck is that? Don't worry, I've got you. Everyone, please imagine a large seafaring vessel, like a massive cargo ship. Long, life-saving story short, inside that boat are tanks dedicated to maintaining stability, its structural integrity, and maneuverability, and to do so, these tanks need to actually take in water to balance everything out. They compensate for weight when a vessel is empty or only partially loaded with cargo, ensuring the ship doesn't capsize and keep the propeller fully submerged. Now, according to the European Maritime Safety website, crews are required to flush and exchange their tanks far out into the open ocean before reaching coastal waters, and are required to use specialized filtration and ultraviolet or chemical treatments to kill or neutralize marine organisms before the water is pumped back out. Sometimes that doesn't happen. And what happens next is a bunch of walnuts in your fjord. What can I say? Put a pin in this. We will get back to it. In the meantime, let's return to our A-team who happened to notice an increased presence of these two drifters the morning after the catastrophic worm orgy, which is a combination of words I never thought I'd say. Is it possible that these creatures, who rely entirely on ocean currents and mild pulsing to swim, are they making their way here deliberately to feed? There's only one way to find out. From their study, quote, field sampling was conducted at two shallow estuarine sites. Site 1, in between Kjørtingfjord and Kjørteminde Fjord, and Site 2 near the mouth of Kjørteminde Fjord. Sampling was conducted during multiple field visits between August of 2022 to July 2023, where A. orida, the moon jelly, and M. ledei, the sea walnut, and free-swimming polychaetes were collected near the water surface using a handheld dip net from a small boat. End quote. My friends, as reported in their study and in their press release, over the course of a year, the researchers recorded 56 cases where the jellyfish contained at least one polychaete in their gut. And if you drop whatever worm you're holding right now and pick up your phone to tap on today's post, you will see a moon jelly with a worm in it. You can't miss it. Moon jellies are basically transparent. There's nothing else inside this one but gonads and a worm. Again, a combination of words I never thought I'd say. Let's take a closer look at those 56 cases instead, shall we? Okay. Among the 166 collected moon jellies, 45 individuals contained at least one polychaete. Among the 71 walnuts, 3 contained at least one polychaete, all during summer months. And the researchers believe the actual number is likely higher. Quote, it takes only a couple of hours for the jellyfish to digest these worms, so there were probably more cases than the ones we observed. End quote, said co-author Professor Eric Kirstensen in the press release. Co-author and Associate Professor Jamila Javed-Pour followed up this by noting, quote, we also suspect that Nemiopsis consumed more worms than we could detect. They also hunt at night, which is exactly when the worms swarm. By the time we sampled them in daylight, the worms could have been fully digested and impossible for us to see. End quote. To confirm that the worms served as actual nutrition and weren't ingested by accident, the A-team went farther than just counting polychaetes. The researchers actually performed an isotope analysis on the jellyfish tissue. And after all the clickety-clack calculations, the results showed that nutrients from the worms were absorbed by the jellies and the jellyfish, rather than, just passing through their transient anuses. Yeah, say it again with me. A combination of words I never thought I'd say. So, what does this all mean exactly? Well, 1, they're fucking hunting. And 2, polychaetes are probably not a major food source, but they do represent, quote, a concentrated seasonal burst of energy in summer that may help jellyfish thrive, end quote, from the press release. And that may be a big problem when it comes to the invasive sea walnut. As the team explained, their discovery not only sheds new light on jellyfish feeding behavior, but also reveals a previously overlooked exchange of energy between the sea floor and the water column. Typically, this flow is seen as one way, organic material sinking down to bottom-dwelling animals, material such as dead polychaetes after spawning. Quote, but here we see energy moving upward instead, bottom-dwelling animals rising into the water column and being hunted there. While several predator species are known to exploit these spawning events, it has not been documented before in jellyfish or Nemiopsis. This means gelatinous zooplankton are tapping into a benthic energy source, or an energy source located at the sea floor, and that has been largely overlooked. we need to consider this upward transfer when modeling the resilience of coastal ecosystems. Even sporadic access to benthic resources may influence how both native and invasive gelatinous species compete and persist. End quote, said Jamila in a concerned tone to Eureka Alert in their press release. So, everyone, please take proper care of your ballast water. You never know if predatory sea nuts may be inside. After the break, we are leaving gelatinous pseudo-fish for actual fish and swapping worms for cocaine. But don't worry, we won't have to travel very far. Just to the north of Denmark, you'll find Sweden. And it's here a first-of-its-kind study has taken place. A wound-up international group of researchers from Griffith University, the Swedish University of Agricultural Sciences, the Zoological Society of London, and Max Planck Institute of Animal Behavior, they have demonstrated the effects of cocaine contamination on fish behavior in the wild rather than in laboratory conditions. And you probably have a lot of questions like, how do you give a fish a bump? And why are we doing this again? All shall be revealed. Please, stay tuned.
I wrote a little song to remind you, Choice Hotels get you more of the experiences you value. The Cambria Hotel's got it all. A rooftop bar, have a ball. Bring a date, your squad, or even your mom. Book direct at choicehotels.com.
Hey, it's Ryan Reynolds here from Mint Mobile. Now, I was looking for fun ways to tell you that Mint's offer of unlimited premium wireless for $15 a month is back. So I thought it would be fun if we made $15 bills. But it turns out that's very illegal. So there goes my big idea for the commercial. Give it a try at mintmobile.com/switch.
Upfront payment of $45 for 3 months, $90 for 6 months, or $180 for 12-month plan required. $15 per month equivalent. Taxes and fees extra. Initial plan term only. Greater than 50 gigabytes may slow when network is busy. See terms.
And we're back. We are so back. And my friends, I'm going to need you to slap on your tightest, finest wetsuit that shows off what you're working with and to join me at the southwest corner of Lake Vättern, which at nearly 2,000 square kilometers or 772 square miles is the second largest lake in Sweden and the largest lake by water volume. Now, that is one fun fact about this lake, so you know what this means. I'm going to drop a few more on you right now. Number 2, it supplies drinking water directly to over 250,000 residents. And number 3, the waters are so clear. At some points, you can look 17 meters or 56 feet straight down. Fun fact number 4. Don't let that clarity fool you. This lake, like so many other bodies of water on our fair planet, has tested positive for cocaine. According to the study we're snorting for this portion of the show, a study revealingly titled, "Cocaine Pollution Alters the Movement and Space Use of Atlantic Salmon in a Large Natural Lake," quote, an analysis of Swedish wastewater affluent within the native distribution of Atlantic salmon revealed maximum discharge rates of approximately 3.6 and 14.1 grams per day of cocaine and benzoylecgonine to aquatic environments, respectively. End quote. My friends, this is a fancy way of saying if you wanted to investigate whether exposure to cocaine and benzoylecgonine, aka the metabolite created when the human liver breaks down cocaine, could affect the movement of Atlantic salmon, and you wanted to do so in a natural environment rather than a lab setting, you just found yourself the lake to run your experiment. Now, if you've joined us during any of the other 5 episodes where we talk about the effects of such substances on animals, then you know that these substances end up being excreted and subsequently detected in aquatic environments due to insufficient removal during wastewater treatment. And in those episodes, and from those highly controlled lab studies, we learned a few things. Like how diluted concentrations of cocaine were shown to accumulate in the tissues of European eels after a 1-month exposure, with the highest levels recorded in the brain, followed by muscle tissue and the liver. If you haven't joined us, and you're wondering how eels get exposed to coke, It's thanks to a lack of toilets at music festivals. I'm serious, you can't make this up. The research is there, and the research says illicit substances are very much physically affecting our watery friends. But, like I mentioned, this has been confirmed and constrained by lab settings. As such, Marcus Michelangeli at Griffith University, ecologists Jack Brand and Michael Bertram of the Swedish University of Agricultural Sciences, and a line full of fellow co-authors wanted to take these kinds of studies to the next level, having animals ride the snake out in the wild blue yonder. To do so, join me, will you, along the shores of Lake Vättern, where over 8 weeks, quote, we experimentally exposed 2-year-old hatchery-reared Atlantic salmon smolts to cocaine or benzoylecgonine in the field using previously validated slow-release chemical implants. Treatment groups, of which there were 35 fish, were administered either a cocaine or a metabolite implant or an implant containing no drug, i.e. the control implant, end quote, from their study published in the April 2026 issue of Current Biology. So yes, you heard me, my outgoing business goose. Young fish between the ages of 1 and 3 are also called smolts. During this time, they are undergoing a major physical and chemical change that prepares them to migrate from freshwater rivers to the salty ocean. In sum, this is the time they are curious about their surroundings, and they have this innate urge to swim that, just like with our marine worms, is bursting at the seams. Please, put a pin in that swimming and drop whatever implant you're holding right now and pick up your phone to check out today's post. You'll see the team's IKEA-like illustration of what these implants looked like. 35 slow-release chemical implants containing the lake's cocaine levels were attached to one group of fish, while 35 others received the metabolite. The remaining 35 dorks got a whole bunch of nothing, but inside all of these instruments was an acoustic tracker that would record each fish's wanderings throughout Vättern. So, after each of the 105 smolts were tagged and released, and after all the pings were recorded, what did the team find? Drumroll, please. Thank you in the back. From the Swedishly named Eric Stockstad of Science.org, quote, in general, young hatchery salmon tend to explore more after they're released. As they learn about their new environment, they move around less and less. This is what the researchers observed over the 2 months. With a twist. Compared with the controls, fish exposed to the metabolite benzoylecgonine swam as much as 1.9 times farther each week. By the end of the experiment, the control fish had largely settled about 20 kilometers or 12 miles from the release site in the south of the lake. Cocaine-exposed fish were living somewhat farther afield, and those exposed to benzoylecgonine had dispersed roughly 32 kilometers or 20 miles from the release point. End quote. Okay, I can see your reaction from here. I think you're thinking, oh my God, it's the metabolite affecting the fish on a greater scale. No pun intended. And it's giving them the jitters? This has to help, right? I am so glad you asked. On face value, that's what it looks like, right? Especially when coupled with this discovery. The average time until 50% of fish from each treatment group were presumed dead was 75 days and 73 days in the cocaine and metabolite treatment groups, compared with 55 days in the control group. This actually may not be a great sign. In an interview with Cymex.org, co-author Dr. Marcus noted, quote, where fish go determines what they eat, what eats them, and how populations are structured. If pollution is changing these patterns, it has the potential to affect ecosystems in ways we are only beginning to understand. End quote. Rachel Ann Hauser-Davis, a biologist at Oswaldo Cruz Foundation in Brazil who wasn't involved in the research, told Scientific American, fish that travel longer distances might enter unsuitable habitats or spend valuable energy on swimming rather than using it to forage and grow. And they might change what types of predators and food they encounter. Now, as to why in the holy hell it's the human metabolite that affects the fish more so than the cocaine itself? Drumroll, please. We're not entirely sure. Yes, and again, thank you in the back. We do know that benzoylecgonine occurs at higher concentrations in waterways, and that it could accumulate more efficiently in the brains of fish. And when you put these two things together, you've got salmon opening up restaurants in parts of town that are doomed to fail. So, what can we do about the situation? Well, better wastewater management that focuses on reduced raw sewage discharges could help lower risks to wildlife, since it's storm overflows and misconnects in household plumbing that are the major sources of the stuff. And if you find yourself at a 3-day EDM festival, for the love of God, stay in line for the Porta-John. You'll be doing our guild friends a favor. And thank you, the rest of you, for listening, rating, subscribing, telling your friends about all the cocaine and all the metabolites in our water, and how it's doing all sorts of fucked up things to fish's brains. They're not going to be expecting this conversation, and they'll be like, I have nothing to do with this, and you'll just look at them silently, and then just tell them about jellyfish and how they can hunt. It's another goddamn thing to worry about in the sea. And a peppy thanks to the folks over at Airwave Media, the podcast network to which WTI belongs. If you love this show, and you do, you'll love the other podcasts in this family. And please, stay interesting.